today's video. The purpose of this video is to demonstrate the process of respiration. Respiration is an important process for biological organisms, including unicellular yeast and bacteria, and multicellular organisms like you and me. This video is going to be broken down into three principal parts. The first part is going to include the definition of life, including the 5 principal characteristics of life. The second part of this video is going to include the definition of respiration and why it is important for biological organisms like you and me. The third part of this video is going to include the use of 3 caustic reagents: bleach, vinegar, also called acetic acid, and hydrogen peroxide to arrest respiration in yeast. All right, let's get to the video. I hope you all enjoy. First, we're going to define the 5 characteristics that define life. The first of these characteristics include responsiveness to the environment. The second of the characteristics include growth and development, or growth and change. The third of these characteristics, as shown up here, is the ability to reproduce. The fourth is metabolism and respiration. This is where we come in. And the fifth is the ability to maintain homeostasis. Without these 5 characteristics, we cannot classify an organism as alive. Second, we're going to discuss the biological process of respiration. Respiration is a biological process for bacteria and yeast, and also other unicellular and multicellular organisms, in which you take an energy source and oxygen, ambient oxygen, and produce energy for the organism. The products of respiration include carbon dioxide and water. The chemical form for this particular video, we're going to be using glucose as our energy source and ambient oxygen to perform this reaction. The chemical form of glucose is represented by the formula C6H12O6. That said, you can also use an isomer of glucose if you like. From the chemical reaction of glucose and the oxygen, the exergonic reaction that occurs, that's a reaction in which energy is released. We have carbon dioxide and water that is formed. In this way, the organism produces energy for itself, and in this way, we show there's a metabolism as one of the primary signs of life. Third, we're going to talk about the 3 caustic materials and 1 control to show how to arrest respiration in the yeast samples we have here today. The first of the 3 materials we're going to use is hydrogen peroxide. Upon exposure to light, hydrogen peroxide produces radicals. These radicals disrupt the glycolytic process in the yeast cells. This results in acidosis in the cell and retards respiration. As a reminder, respiration is a key aspect, a key characteristic of life. The second 2 caustic materials we're going to use are both acids, and they're both weak acids. The first of these 2 is acetic acid, which functions to basically produce hydrogen ions in the solution. These reduce the pH, denature the enzymes, and also retard respiration. The second of the compounds we're going to use is called hypochlorite acid. This is commonly created with bleach. Bleach uses sodium hypochlorite, and upon it going into water, you can have hypochlorite acid formed. Again, similar to acetic acid, which is vinegar, the hypochlorite acid creates hydrogen ions. These reduce the pH, and ultimately, these arrest respiration. As a reminder, respiration is a characteristic of life. Thus, when you add these compounds, they can kill the yeast that's present. All right, for our caustic materials, we're going to be using 3 different materials. Our 2 acids are going to be acetic acid and our bleach. Now remember, they're both weak acids and they have different acid dissociation constants. That said, where we'll be using household bleach, which has a roughly 5 to 6% sodium hypochlorite, we're going to be using industrial strength acetic acid, which has roughly 75% volume for volume acetic acid. Lastly, we're also going to be using our source of radicals, and that is our pharmacy grade hydrogen peroxide. This is 3% volume for volume hydrogen peroxide. Now, can you guess which of these are going to have the greatest effect on our yeast samples? All right, let's test and see. To begin this experiment, we're going to start with 30 cc's of sugar, 120 milliliters of water, and 10 cc's of dry active yeast. The waters have been colored to indicate the different caustic reagents that will be added to them later. That said, in principle, they all will start the same with yeast, sugar, and water. To encourage respiration, the yeast sugar mixture shall be mixed to move around the sugar and to allow for the yeast to begin the fermentation, to begin the respiration process. Furthermore, to indicate that respiration is occurring, the following balloons will be placed upon each sample, with the goal of capturing the released carbon dioxide as the yeast perform respiration. We'll allow these to rise for the remaining 5 to 10 minutes. Then we'll come back and add our caustic materials to the 3 experimental vials. We're several minutes later, and as we can see, the yeast has performed respiration, as evidenced by the presence of the carbon dioxide that are trapped in these balloons. Basically, the yeast are processing the glucose, the products of that are carbon dioxide and water, as we expected. Now, with these 3 vials, we're going to add our 3 caustic reagents, respectively, to each vial. We're going to see what effects these have on the yeast, and if they do indeed arrest respiration. To each respective vial, we're going to add roughly 4 milliliters of our given caustic reagents. That said, first, I'm going to remove the balloons from all of the cases so that we have uniformity. First, we're going to add 4 milliliters of hydrogen peroxide. Remember what I explained. Hydrogen peroxide becomes activated once exposed to light. Next, we'll add 4 milliliters of acetic acid. In this particular case, we're using 75% volume by volume concentrated acetic acid. Therefore, we'll use safety first. You ready? Last, we'll use 4 milliliters of household bleach. As a reminder, safety first. Now we're gonna mix all the vials. And now we're going to see the effect that this has on the creation of more carbon dioxide. We'll leave this for several more minutes. And our goal is to see how much our balloons indeed inflate. Alright, so we have several minutes later, and we can see that despite the fact we added 5 milliliters of our hydrogen peroxide, we still have growth going on there. So it's expected. The radicals damaged the cells, but they didn't necessarily destroy them. We have a whole different situation here with our acetic acid. We had 75% volume by volume acetic acid added to this vial. As you can see, the yeast has completely died. There are no more any carbon dioxide bubbles, and even the balloon won't inflate. So we have arrested respiration in this case. Moving on to the solution in which we added 5 to 6% sodium hypochlorite. As you remember, both acetic acid and hypochlorous acid are weak acids. That said, it's not just the strength of the acid that's important, but the concentration. In this case, our concentration was so weak that some of the yeast continued to live, resulting in respiration, and thus we did not arrest respiration. So there we have it, 3 different cases, and the control, which is doing better than ever. I want you to compare the size of the balloons for the control, hydrogen peroxide, the acetic acid, and the bleach. As you can see, as we get, in the case of the worst case, we have no inflation. But as we get worse with our caustic reagents, our inflation, our bubbles become smaller. Did you guess the right answer? Let me know in the comments below.